Hello students, today we will start about new topic, that is about mutations. Mutations are defined as sudden heritable chromosomal changes or genetical changes. Those heritable changes, they can be either physical changes or chemical changes in the genes or chromosomes. These mutations are first discovered by Hugo de Vries in the plant Enothera lamarckiana. Now, what is the chance of mutation normally naturally in a cell? Naturally, mutation can happen only in one cell out of 10 lakh million number of cells. That we can write with a term, that is mutation frequency. Mutation frequency in a natural in a normal cell is one cell can be exposed to mutation out of 10 power 6 million number of cells. So normally there is no chance, but if a mutation has to happen, it will happen only in one number of cell out of this many number of cells. Now, this is about natural mutation. Then there are some other types of mutations that are happening because of some chemicals or some radiations. So we can say that the substance that there are some substances that are causing mutations. So what is name of those substances? The substances that cause mutations are called as mutagens. There are different types of mutagens. Some are chemical mutagens and some are physical mutagens. So under chemical mutagens, we can see some of the chemicals. Under physical mutagens, we can see some of the radiations that are exposed either by X-rays or gamma rays or alpha rays, etc. That also causes mutations. Mutagens are of two types. One is chemical mutagens, physical mutagens. Now, chemical mutagens, those are nothing but chemicals. So, examples like chemicals called, first one, 5-bromouracil. Second example, 5-iodouracil. This is indicated as 5 capital B and U. This is indicated as 5 capital I and U. Now, these both can be called as 5-bromouracil can be called as base analog of 5-methylated uracil, that is thymine. So this is called as base analog of thymine. What is thymine? Thymine is one of the nitrogen base out of 4 different types of nitrogen bases in DNA. Uracil is present in RNA. That comes under out of 4 different types of nitrogen bases in RNA. So second mutagen, chemical mutagen, we can take example as alkylating agents. Alkylating agents. Now, these alkylating agents are some of the drugs we can consider or the chemicals that are used during cancer treatment. They bind to the 7th position of a nitrogen in case of guanine. What are these alkylating agents? Are drugs used in cancer treatment. Another type of chemical mutagen is acridine dyes. Acridine dyes, they are also some types of chemical substances that are responsible for frame shift type of mutations. Causes frame shift mutations. Next, another type of mutagen or chemical mutagen is mustard gas. Mustard gas is considered as the first identified chemical mutagen. Another example is also there under chemical mutagens, that is nitrous acid, HNO2. So fifth example is nitrous acid. These are few number of chemical mutagens that causes mutations. We will see what are physical mutagens also. First one, ionizing radiations. These type of radiations converts even atom-sized particles also into ions. So, under example of ionizing radiations, we can take alpha rays, beta rays, gamma rays. Next, X-rays also we can consider under ionizing type of radiations. As I have said you, they causes ionizing of even very much small sized particle like atom also. Next, non-ionizing radiations. Non-ionizing radiation. Here examples are UV rays or UV radiations. We can also use term like UV radiation. Now, what is the main danger about these UV radiations or UV ultraviolet rays? These causes thymine dimers. Thymine dimers means usually in one, if we think this is the double-stranded DNA, we think that is double-stranded DNA, and one end is like this, second end or second strand is like this. Then the sequence should be adenine, thymine, guanine, thymine, thymine, or like this the sequence has to be. Between, okay, so there will be hydrogen bonds which are complementary to another type of nitrogen base on another strand like this. For guanine means it will be triple bond cytosine, etc. Between these two, one nitrogen base and another nitrogen base, there should be diphosphoester bond that is connecting two side by side nitrogen bases. So here should be diphosphoester bonds. But instead of that, between two thymines means two nitrogen bases on a single strand, there occurs covalent bond. Means, again I am repeating, between two side by side thymine nitrogen bases on one single strand, there should be diphosphoester bond. But instead of that, there occurs covalent bond between two side by side thymines on a single strand. That we call as thymine dimers. Okay, so this is the severity of physical and chemical mutagens that are causing. Now we will see what are the types of mutations basing on the changes that they are causing in the morphological structure of the chromosome or inside the genetic arrangement of the chromosomes. Types of mutations. There are two types of mutations. We can say one is chromosomal mutations. Second type of mutations are point mutations. So, if mutations are happening by changing the structure of the chromosome, that is considered as morphological changes. But if there is any change in the number of chromosomes, then it is called as numerical mutations. Now, mutations that happen on a part of chromosome or in or change in number of chromosomes. It can be change in number of either sets also. Those are chromosomal mutations. Means here we can see either change in the structure of chromosome also. Again, these chromosomal mutations are of two types. One is morphological mutations. You may ask me, why should we read all this? Because there are previous MCQs that are asked on translocation type of mutations, and some options are given like deletion mutations and duplication mutations, etc. in previous MCQs that are conducted after 2018. So we need to discuss about this type of chromosomal mutations. Morphological mutations, and another one is numerical mutations. Now, under morphological mutations, again there are few types of mutations. They are named as deletion mutation or duplication type of mutations, inversion mutations, translocation mutations, reciprocal type of mutations. Whatever the disorders that are caused by such type of mutations, they are also given in our NCERT textbook, like Down's syndrome, thalassemia, etc. So we need to learn about these type of mutations also. So please don't neglect to learn. Types of morphological mutations. First one is deletion mutation. If in an chromosome, within the chromosome, if any type of or any number of genes are deleted or broken, means one gene is suddenly absent inside the chromosome, then such type of mutations are called as deletion mutations. So we can represent this one in a diagrammatic form like this. For suppose there are genes like A, B, C, and in another chromatid part there are some other type of genes like 1, 2, 3. Suddenly this second and third genes are deleted or disappeared in the same chromosome. We are not considering between homologous or heterologous type of chromosomes. In a chromosome, if suddenly the chromosome has lost some genes inside it, lost means they are disappeared or broken into particles, then such type of condition is called as deletion mutation. If some genes are lost within a chromosome, then such type of mutation is called as deletion mutation. Here, what type of disorder can be considered in such cases? Example we can take as thalassemia. Thalassemia is a blood disorder in which oxygen carrying hemoglobin is decreased to very less number. Is a blood disorder in which oxygen carrying hemoglobin, I am indicating hemoglobin as capital H small b, is decreased to more extent. So this is one example under morphological mutations. Another example we'll see. Second one, inversion type of mutations. Now, if the genes within a chromosome are means translocated, means flipped from one place to another place, or changed their position from one place to another place within a chromosome, then such type of mutations are called as inversion type of mutations. For suppose, I'll read the sentence which means of inversion mutations. If genes within a chromosome are change their positions then such type of mutations are called as inversion type of mutation. I am not considering between homologous or non-homologous, just I am taking one chromosome and showing the diagrammatic representation of inversion type of mutations. Again, we will take the same type. Some genes, suppose ABC type of genes are present in a chromosome. Here on another chromatid or arm, there are 1, 2, 3 type of mutations. Now, one has to be present near to the centromere, 3 has to be present away from the centromere. It is a normal condition for supposing. If the third gene has been shifted to first gene position and first one has replaced that third position away from the centromere, such type of mutation is called as inversion type of mutations. So, then the shape of the genes inside the position of the genes inside the chromosome will be changed. In this, it will be same. We can also write by shifting C to A and A to C positions. Here we can also say they are changing in the sense completely the loci is being changing its position. So it will be 3, 2, 1. Means here in case of inversion mutation, the loci is completely changing its position. Now, I did not tell you example under inversion type of mutations. Let's take one example. Inversion mutation. One example is hemophilia A type of disorder. Hemophilia A type of disorder. This type of disorder is considered as X-linked disorder. X-linked disorder in blood clotting factor 8. Blood clotting factor 8 gene. If any such type of gene has changed its location or locus in a chromosome, then it leads to hemophilia A. That gene has to be blood clotting factor 8 protein coding gene. Then it causes hemophilia A. This one we can take under example of inversion type of mutations. Third example we can take duplicating mutations or duplication mutations. Now, within a chromosome, again, if any number of genes are doubled or duplicated, if any gene on a chromosome are duplicated or doubled, this we have to read within a chromosome. Within a chromosome. Now, so diagrammatically we can represent, again we will take the same types of genes. If the first gene, second gene which are present on different loci are doubled, then how the chromosome is going to change? Its morphological structure has to be changed and the size of chromosome also has to be increased. So here it will be same. If these both are duplicated, then 1 and 2 number of loci or genes will be there. Again, since they have been duplicated, another set of same one gene, second gene also will be present. The third one will be as this. So we can consider these as the duplicated genes. So what are the disorders that we can read under duplication? Here we can take one disorder as an example, that is Bar-Eye syndrome. In case of Bar-Eye syndrome, eyes become narrow. They become like this. Their shape, actually our face will be having like this horizontally, but their eyes will be like this in this position. So, eyes become narrow. So, these are the two, third example. This is about duplication mutation. Next. In case of translocation mutation, it is very important to learn that this type of mutation happens between non-homologous chromosomes. Means which are not having identical genetic material or which are not means present in identical gametes. Here, the genes are shifted only in one direction. The gene transfer is in one direction between non-homologous chromosomes. If you see this same sentence in diagrammatic form, if you think that these both are not homologous chromosomes, not going to pay, but they are side by side. Now, take out the genes C, D, E, means different types of genes in different locus of the chromosomes, means different positions on the chromosome. 1, 2, 3, 4, 5, 6. As I have given here sentence, the gene transfer is in one direction. Means some of the genes from one chromosome, they are shifted or translocated to another non-homologous chromosomes. For suppose here, okay, B has been translocated to third position of the non-homologous pair of chromosome. E has been shifted to fifth chromosomal place. Then how it is going to look? The first chromosome, which is this one, it is having A now, B place is empty, and C type of genes. Now here in the second part of the chromosome, there will be D and next will be F, away from the centromere. Now, the non-homologous pair chromosome will be as it is, but the genes will be more. Means the formation of loci also will be more in the second part of the non-homologous pair of chromosomes. So let's write here what are the genes. Here it will be 1, 2, B has come to third place, so B, 3. The number of genes and the loci are increased in the second part or second chromosome of the non-homologous pair. Now, what about here? It will be 4th. So E has shifted here 5 and 6. So the number of genes in the chromosome has been shifted. Now, what example can we take here to learn about this type of mutation? We can take Down's syndrome. Down's syndrome example is in NCERT textbook. So, we should learn about this one in detail. Sorry. Reciprocal type of mutation. Now, this also happens between non-homologous chromosomes. We have seen in case of translocation type of mutation, genes are getting transferred in one direction, means from one chromosome of non-homologous pair to another chromosome of non-homologous pair. But in case of reciprocal, both the chromosomes exchange the genes between them. So here between non-homologous pair of chromosomes, genes are transferred in two directions. Transferred, we can also use a term like exchange. Between each other. This sentence we can use easily to understand reciprocal type of mutation. So if you see the diagrammatic representation between two non-homologous type of chromosomes, let's give out the gene naming as C, D, E. Here 1, 2, 3, 4, 5, 6. Just for our understanding, I am giving the gene names like this. If 4 is shifted to third position, D can also shift to fifth position. If B can shift to second position, third chromosome can shift to C position. So after reciprocal type of mutation, the arrangement of genes are going to be A will be as it is, but B is replaced by 2 and C is replaced by 3. In case of this one, here it will be 4, 5 and E. Now, if D is shifting to fifth position, then fifth chromosome can easily shift to D position. Okay. So we will write here 1, B, C, and here C, D, 6. So this will be the type of arrangement after reciprocal mutations. Now, the chromosomes in such type of reciprocal type of mutations are given a term like this type of reciprocal mutations happens between what type of chromosomes? If a question was asked, answer will be the chromosomes are termed as Philadelphia chromosomes. These non-homologous chromosomes which are responsible for reciprocal mutations are termed as Philadelphia chromosomes. These Philadelphia type of chromosomes mostly will be 22nd chromosome. 22nd, we can also use the term like 22nd pair of chromosomes. And even 9th chromosome also we can take as Philadelphia chromosome. Now, what type of disorder these Philadelphia type of non-homologous chromosomes causes? They causes chronic myeloid leukemia, which we indicate as capital C, capital M and capital L. This is abbreviated as chronic myeloid leukemia. This also can cause another type of disorder in case of bone marrow, that is lymphoblastic leukemia, acute lymphoblastic leukemia, which we give a shortcut like capital E, capital L and capital L. This is acute lymphoblastic leukemia. Acute lymphoblastic leukemia, which we are representing with all ALL, but all the letters are capital form. So this is about chromosomal morphological mutations. There is another type of mutation, that is numerical mutation, which comes under chromosomal mutations. Here in case of numerical mutations, there are again of two types, that is change in number of sets of chromosomes and also change in total number of chromosomes. If the change in number of sets of chromosomes happen, then it is called as euploidy type of numerical mutation. And if there are changes in total number of chromosomes, then such type of mutations are called as aneuploidy type of mutations. We will see about complete details about numerical mutations in the next video.